Good day, everyone. Before I begin, I would like to introduce myself as the presenter. I am Mary Rana D. Sumaya, and I am here to tackle about deoxyribonucleic acid, also known as DNA. I will discuss the DNA structures and its step-by-step replication process. To begin with, let's first understand the structure of DNA. DNA. What does it mean? The D in DNA stands for deoxyribo, N for nucleic, and A for acid. DNA is an organic compound that belongs to the four macromolecules, which is the nucleic acid. It contains two strands, the double helix strand that is composed of repeating subunit called nucleotide. In general, there are three basic components of nucleotide. First, we have here pentose sugar or 5-carbon sugar. So we have first carbon, second carbon, third, fourth, and fifth carbon. Second is the nitrogen base. As an example, we have here adenine, and then lastly, the phosphate group. So this is the phosphate group. What makes DNA nucleotide? Among DNA, there are four nitrogen bases. We have adenine, guanine, thymine, and cytosine. These four nitrogenous bases are classified according to their structure, which is the presence of aromatic rings or the number of aromatic rings. So adenine and guanine has two aromatic rings, whereas thymine and cytosine has one aromatic rings. Adenine and guanine having two aromatic rings are then classified as purine, while thymine and cytosine are then classified as pyrimidine, that it only has one, single aromatic ring. So this is the nucleotide pairing. Adenine is double bonded to thymine through hydrogen bonds, while cytosine is triple bonded to guanine through hydrogen bonds as well. Let's proceed to the organization of DNA nucleotide components. So the nitrogen base is connected to the first carbon sugar of the ribose sugar through the N-glycosidic bond. On the other hand, phosphate group is connected to the fifth carbon of the pentose sugar using an acid. So this slide will tell us how these components organized into DNA molecule. But first, how are these components organized? The answer is through the formation of hydrogen bonds between nitrogen bases. For example, we have here cytosine and then the guanine. There are three hydrogen bonds in between. Even though hydrogen bases are weak, but in numbers, they are strong bonds. That is why they can hold two strands of DNA molecule together. Another property of DNA molecule is that each strand runs in opposite direction, meaning each strand has opposite polarity. The other strand runs from 5' to 5' direction, while the other strand runs from 3' to 5', but in opposite direction. Since we have the knowledge about DNA structure, we can now proceed to the DNA replication and its step-by-step process. First, we will break down the replication into 4 steps. We have here the initiation as the first step. Second is the unwinding. Third is the elongation. Fourth is the termination. And first, we're gonna take a look of replication in the context of using prokaryotic model. Because in prokaryotes, genetics are a little bit simpler than eukaryotes. Prokaryotic replication is going to begin at a single origin of replication. So, the first step is initiation. Initiation occurs if a sufficient sufficient source are available to complete a round of DNA replication. This involves the origin of replication and initiator proteins. Then, the initiator protein binds the origin of replication, creating tension in the adjacent nucleotide. The tension in the adjacent nucleotide disrupts the hydrogen bonds between the pair and it makes the DNA molecule slightly opens up. During replication, DNA helicases unwind DNA at position called origins where synthesis will be initiated. Excuse me. Helicase open helix and binds primase to form primose. RNA synthesis enables DNA polymerase to start first DNA chain. As DNA helicase continues to unwind, the DNA will form a structure called replication fork. So, the third step of DNA replication is the elongation. Elongation is a step in which the DNA synthesis is initiated. But before the start of elongation, primase synthesized RNA primer. The DNA polymerase cannot able to add nucleotide without an RNA primer because it provides a start site as a free 3' negative OH group for the polymerase to work. So this is the enlarged image of primase. The first step of elongation is the DNA polymerase. There are 3 different types of DNA polymerase that helps in prokaryotic DNA replication. And these are the following. DNA polymerase 1 removes RNA primer by exonuclease activity and proofreads the DNA replication. The DNA polymerase 2 is the repair function. The DNA polymerase 2 mainly involved in the DNA repair pathway, while the DNA polymerase 3 adds nucleotide from 5' to 3' direction. Next is we will tackle the activity of the DNA polymerase. These 2 DNA polymerase activity are the following. First is the leading strand synthesis, a 3' negative OH end of single-stranded DNA. It is a very straightforward synthesis and starts at primosome and continuously adds nucleotides until termination sequences. Next is the lagging strand. Lagging strands synthesis in which it has the 5' negative P end. Hence, the polymerase cannot synthesize new DNA from this direction. In here, primase synthesizes multiple primers and synthesized in short DNA fragments called Okazaki fragment. SSBP or the single-stranded binding protein helps the single-stranded DNA from the attack of nuclease. It is present throughout the replication process to protect the DNA's stranded DNA. After the synthesis of leading and lagging strand, The polymerase is detached from the site of replication. And now we are on the fourth or last step of replication process, which is the termination. On stage 1, the multiple primers at lagging strand are cleaved and removed by DNA polymerase 1. It also fills the gap between two Okazaki fragments by the addition of nucleotides. Furthermore, DNA polymerase proofreads the sequences for avoiding error in replication. On this stage, the enzyme DNA ligase fills the gap and creates a photodiester bond between Okazaki fragments and newly added nucleotides. At the last stage of termination, two replication fork meets a terminator recognizing sequences called TER. TER stands for DNA replication terminus. TER sequence with TUS protein. TUS means terminus utilization substance, and this protein creates a complex which arrests the replication fork and prevent them to escape. Only DNA topoisomerase 2 remains in action. It cuts both strands, dissociates TER to TUS complex, and then the two different circular DNA is generated. So that ends with my report. Thank you for listening. God bless.